Hi, everyone. My name is Shawn Gu from the University of Notre Dame. Today, I'm going to talk about our work modeling multi-scale data via a network of networks. As I'm sure we all know, understanding the functions of human proteins is important. One way we can study human proteins and their functions is with a protein-protein interaction network, or PPI network. In such a network, nodes are proteins and edges are PPIs. Then, we can apply network science methods that deal with the task of node label prediction. Given a PPI network, the goal is to predict functions or labels of proteins or nodes. In a complementary fashion, we can also analyze a protein's 3D structure to understand its function. In particular, a protein's 3D structure is often modeled using a protein structure network, or PSN. In a PSN, nodes are amino acids of a given protein, and edges join amino acids close in the 3D space of the folded protein. Then, we can apply network science methods that deal with the task of graph label prediction. Given multiple proteins PSNs, or graphs, the goal is to predict their functions, or labels. So sometimes, the entities, in this case proteins, modeled by nodes in a network, in this case a PPI network, can themselves be modeled as networks, in this case PSNs. So we can integrate this multi-scale or multi-level data into a network of networks, or NON. I've represented that visually here, where a node in the higher level PPI network itself corresponds to a protein structure network at the lower level. More generally, we call the higher level network the level 2 network, and each lower level network a level 1 network. Here, I've only shown 3 level 2 nodes to have corresponding level 1 networks, but in general, every level 2 node can have a corresponding level 1 network. An important aspect of our definition is that given any two different proteins and their PSNs, none of the nodes overlap. To give some intuition into why we impose this condition, suppose we have two amino acids from different proteins. These amino acids are not the same physical entity, even if the type of amino acid is the same. So, when we have multiple PSNs, it makes sense that none of their nodes overlap. Then, we can ask, is entity label prediction, in the previous example, this would correspond to predicting labels or functions of proteins, using multiple levels as an NON, more accurate than entity label prediction using a single level alone? Basically, we compare node label prediction using only the level 2 network to graph label prediction using the level 1 networks to entity label prediction using the NON. To emphasize the novelty of our work a little more, to our knowledge, NONs have not yet been studied in the context of entity label prediction. So, we need to develop a framework for evaluating NON-based entity label prediction versus the single level label prediction tasks described previously. It's important to note that some studies analyzing NONs do exist, but they differ in terms of the data analyzed, the application domain, and/or the network science task. So our study is not really comparable to those, and vice versa. Also, some studies do use the term network of networks, but they generally mean a different type of network model. For example, multiplex or interdependent or similar. Here is an outline of our framework. To our knowledge, NON data is scarce. So we develop a synthetic NON generator that can create NONs with various network structural properties. We also construct and provide a biological NON, illustrated previously. Of course, this data is labeled, which we will talk about shortly. Then we test recent state-of-the-art label prediction approaches on this data and evaluate using several measures. First, we'll talk about the synthetic NON generator. We want to generate NONs such that only a label prediction approach using both levels can achieve high label prediction accuracy. So we design our synthetic NON generator to combine instances of two existing single-level random graph models, geometric, or geo, and scale-free, or SF. We choose these two in particular because both have been shown to model PPI networks well, and geo has been shown to model PSNs well. So these are among the most realistic random graph models we could have chosen. Then we label each entity, i.e. level 2 node and its level 1 network, based on the combination of geo and SF it is involved in at the two levels. Here is a toy example. First, we have dotted circles, which contain level 2 nodes that are connected with either a geo or an SF-like network topology. Then, each level 2 node itself has an indication of whether its corresponding level 1 network is of type geo or of type SF. With two models, there are four possible combinations of level 1 and level 2. So we label each entity based on these combinations. Because we have four labels with an equal number of entities per label, we perform balanced multi-class classification. We also vary two additional properties, the across-edge amount and the rewire noise amount. For the across-edge amount, for each of these level 2 node groups, we randomly delete some percentage of edges and then add the same number of edges to go across the node groups. For rewire noise amount, for each of these level 2 node groups, we randomly delete some percentage of edges, but then we add the same number of edges within that same group. Also, for each level 1 network, we randomly delete some percentage of edges and add the same number back. Essentially, as a cross-edge amount increases, the amount of clustering structure in the level 2 network decreases, since more and more edges will be going across these groups. The level 1 networks will not be affected. As rewire noise amount increases, essentially the data is becoming more noisy overall. We also test a real-world biological NON as described previously. Here, we label proteins based on gene ontology data, or GO data. Specifically, we look at protein GO term annotations. After filtering steps, there are 131 GO terms total that we consider. For each GO term, the goal is to predict whether or not each protein is annotated by that GO term. On this data, we evaluate prominent state-of-the-art approaches for node label prediction and graph label prediction, and then we combine these into their NON counterparts. We also propose a graph neural network framework to perform entity label prediction directly on NONs. Also, important thing to note is that these approaches come from either the graph theoretic or graph learning domains, since these two domains are the main ones that study label prediction involving nodes or graphs. Finally, we evaluate each approach in their respective task using a variety of measures. For the synthetic NONs, because the class sizes are balanced, we just evaluate using accuracy. For the biological NONs, however, because class sizes are imbalanced, we consider area under the precision recall curve, precision, recall, and F-score. Now I'll walk through some results. I'm going to start by talking about the synthetic NON, where the across-edge amount and rewire noise amount are small. This sort of serves as our control case. First, we expect random classification to reach 25% accuracy. This is because there are 4 total labels, so 25 corresponds to 1 out of 4. We also expect single level approaches to reach 50% accuracy. Essentially, a single level approach can distinguish between geo and SF at one level, but not at the other level. So essentially, it's only capturing 50% of the information. And we expect NON approaches to reach 100% accuracy because that's how we designed the NON. Here are detailed results for the case when both the across edge amount and rewire noise amount are low. The x-axis contains the different approaches we consider with the NON approaches underlined, and accuracies are listed above. I've roughly separated them into approaches from the graph theoretic domain, graph learning domain, and then our GNN approaches. There's also a bar corresponding to combine all, which is when we integrate information from the two domains. And the methods in combine all are listed here. In the interest of time, I won't be able to go over all the categories in detail. Instead, I'll focus on how NON approaches in general compare to single level ones. Here we see all the expected trends, except L2 sign performs as well as NON approaches, when it should be attaining 50% accuracy. We found this is likely because L2 sign captures clustering structure in the level 2 network. Basically, we looked at L2 sign when the across edge amount and rewire noise amount varied. Remember, these parameters can control how much clustering structure there is in the NON, or how noisy the NON is overall. We found that L2 sign performed well as long as there was clustering structure, regardless of how much noise there was. And this suggested that L2 sign was picking up on that clustering structure. Next, we'll just look at 2 instances of when we increase these parameters. Again, the data is becoming more noisy and the clustering structure is being reduced, so we expect accuracies to drop overall. We do see these trends. At a higher value of these 2 parameters, so 75%, the accuracies are lower. We also see L2 sign doing worse than NON approaches, as the clustering structure is no longer as prominent. This last panel serves as a sanity check. When we maximize the across edge amount and rewire noise amount, we essentially have completely random data. So all approaches are expected to reach 25% accuracy, which is what we see. In total, we tested 30 different parameter combinations of across edge amount and rewire noise amount, corresponding to 30 different NONs. In summary, we found that for 30 out of the 30 NONs, at least one NON approach outperformed or tied the best single-level approach. This indicates that NON approaches are useful on this kind of data. For results on a biological NON, we summarize the performance of each approach over the 131 Go terms. Here, we only show area under precision recall, as results for other measures are qualitatively similar. The x-axis contains the different approaches we consider, and the y-axis indicates the proportion of all Go terms, all 131 Go terms, for which a given approach achieves a certain rank. For example, for combine all, looking at the dark blue bar, it achieves rank 1 for around 20% of all 131 Go terms. That is, for those Go terms, combine all is the best performing approach. Overall, this is just showing that we find NON approaches to be the best for some, but not all Go terms. Looking deeper, there are 69 or 53% of the 131 Go terms where at least one NON approach either outperforms all single-level approaches or at least ties the best single-level approach for at least one evaluation measure. Perhaps more interestingly, we find that for 38 or 29% of all Go terms, only NON approaches perform significantly better than random. That is, for these Go terms, NON approaches make meaningful protein functional predictions, while single-level ones achieve random accuracy. As such, NON approaches are needed, and continued research into NON-based data integration is promising. Finally, To give a brief summary of the running time results, we found that the best NON approaches are, at worst, around 20 times slower than the best single level approaches. And the scale we're looking at is around 10 minutes versus around 30 seconds. So even though NON approaches are slower, they are still computationally feasible on our data. So in this case, we believe the trade-off between accuracy and running time is worth it. But of course, developing efficient NON approaches is another important research direction. With that, I'd like to wrap up. I've listed some references here, including to our paper and to the approaches we compare against. Of course, I'd like to give a shout out to the research group I'm part of, the Complex Networks Lab, and acknowledge the funding agencies. Thank you all for listening.